This presentation is on a research paper titled Modulation of Beta-catenin Signaling by Glucagon Receptor Activation. The glucagon receptor, GCGR, is a member of the class B G-protein coupled family. Glucagon is a peptide hormone secreted by alpha cells of the islets of Langerhans in the pancreas to increase glucose production by stimulation of glucogenolysis and gluconeogenesis. The binding of glucagon to its receptors activates the cyclic adenosine monophosphate (CAMP), protein kinase A, protein kinase C, and mitogen-activated protein kinase pathways. Glucagon is very important for the regulation of glucose homeostasis. Beta-catenin is also a protein that is encoded by the CTN-MB1 gene in humans. It is known as the cadherin-associated protein as it is a subunit of the cadherin protein complex. This research also examined the role of low-density lipoprotein receptor-related protein 5, referred to as LRP5, in glucagon-induced beta-catenin signaling, as LRP5 is an essential co-receptor required for WNT protein-mediated beta-catenin signaling.
The first step in this research experiment was the use of the CRE Luck reporter, that is, Signal Camp Response Element Luck reporter. It is used to monitor the activity of cyclic adenosine monophosphate and protein kinase A signal transduction pathways in cultured cells. To measure CRE activity, human embryonic kidney 293 cells, also known as HEK 293 cells, were transfected with CRE Luck DNA. Cells were plated 24 hours before transfection, and after transfection the cells were induced with reagents. 17 hours after the induction of the reagents, the cells were harvested and lysed in the plate, using 100 microliters of passive lysis buffer at room temperature for 15 minutes. Firefly and Ranilla luciferase activities were measured on an Envision luminometer with a dual luciferase assay kit. Analysis was then carried out. Following the analysis of the luciferase assay, Western blot, also known as protein immunoblot, was carried out. This is a technique used to detect specific proteins in a sample of tissue or extract. HEK293 cells were cultured in 6 well plates. The cells were transfected with DNA using lipofectamine 2000. After transfection with DNA, the cells were then transfected with different reagents. Cell pellets were lysed in 9 microliters of cell lysis buffer on ice for 30 minutes. Western blots were then performed. The blots were probed with anti-beta-catenin antibody and then stripped and reprobed for anti-beta-catenin antibody as a control. The next step was immunoprecipitation, which is the process of precipitating a protein antigen out of solution using an antibody that binds specifically to a particular protein. The antibody binds to the protein, allowing it to be pulled out of the protein complex for further analysis. Immunoprecipitation with V5 antibody was carried out using the antibody extracted from mice. 50 microliters of anti-V5 agarose beads were directly incubated with the cell lysate for 2 hours at 4 degrees Celsius. Mixing was induced by rotating the tube. The beads were spun down and washed 3 times with tris buffered saline with 0.5% tween 20 buffer. 50 to 60 microliters of 2 times reducing electrophoresis buffer was added to the beads and incubated at 37 degrees Celsius for 5 minutes before being loaded onto an SDS gel for analysis. A BRET assay, also known as bioluminescence resonance energy transfer, was performed. This technique allows monitoring of protein-to-protein interactions in real time in living cells. Using COS1 cells, the studies were carried out. The cells were transfected with a fixed concentration of RLU tag constructs for the donor and increasing concentrations of YFP tag constructs as acceptors. After 48 to 72 hours of incubation, the results were analysed.
Figure 1 shows the results from the HEK293 cells that have been cultured in a 24-well plate using CRE Luck reporter. The graph plots HEK293 cells that have been transfected with just CRE Luck and those that have been transfected with both CRE Luck and glucagon receptors against the CRE Luck activity. It shows the cells transfected with glucagon receptors showed a response to the GCG129 agonist, but not to the GCG929 antagonist. The cells were also treated with FSK as a control. It was seen that the FSK activated the CRE luciferase activity independent of the glucagon receptor expression. This proves that glucagon agonist activates the CRA lock activity in glucagon receptor expressing cells. Using the Western blot, it was confirmed that the HEK293 cells have no detectable expression of glucagon receptor until after transfection with a glucagon receptor expression plasmid. This can be seen in column 1, where there are no bands, whereas after the cells have been transfected, distinctive bands can be seen in column 2 and 3. Also in column 4, it can be seen that the primary hepatocytes, which have been isolated from BL/6 mice, have a glucagon receptors. Both results from figure 1 and 2 show that glucagon agonists induce the protein kinase A pathway in glucagon receptor expressing cells. Figure 3 shows that the LRP5 physically interacted with glucagon receptor. This was achieved by the use of immunoprecipitation. This is seen in the wells where there are 2 bands for LRP5 and glucagon receptor. In the first well of LRP5-V5, glucagon receptor without transfected LRP5 showed no expression. Figure 4 shows results gotten when a Brett assay was used to further confirm that ectopic exposed glucagon receptor and low-density lipoprotein receptor-related protein 5 do interact specifically on the cell surface.
In conclusion to the research, it was found that the activation of the glucagon receptor led to the activation of the cyclic adenosine monophosphate and protein kinase A pathway and beta-catenin signaling. Therefore, activation of the beta-catenin signaling is pKa dependent. Additionally, LRP5 physically interacted with the glucagon receptor by immunoprecipitation and Brett assay. These results revealed an unexpected crosstalk between glucagon and beta-catenin signaling. Thank you for listening to our presentation.